So today we'll start with infectious laryngotracheitis. This is also known as ILT. So ILT is a oil-listed disease. This ILT is an acute, highly contagious disease of hens and pheasants characterized by respiratory distress, coughing, expectorations of blood, mucoid exudate, as well as in some cases they are responsible for a very high mortality. This particular ILT disease is caused by Gallid herpesvirus 1. Now we will see the etiology. So etiology, this particular ILT is caused by Gallid herpesvirus type 1. This Gallid herpesvirus type 1 is a double stranded DNA virus, belongs to genus Iltovirus, means they are ILT like viruses, and belongs to family Herpesviridae. As we all aware that herpes virus are responsible for latency, and in this case also this particular virus are responsible for production of latency in infected birds. We have already discussed what is latency. Latency means this particular herpes virus will initially replicate at the target sites like respiratory and nasal epithelium in this case, and when this immunity is developed, it will go into their target, their hiding site, particularly neurons in trigeminal ganglion. So it will infect the neurons, it will go inside the neurons, it will insert their genomic material into the host cells, but there will be no any replications of a virus inside the cells. There will be no any production of viral protein. They will remain silent for a particular period until when their immunity is very high. And whenever there is immunosuppressions, whenever animals or birds, whenever they are in stress, in that conditions, this particular virus will again get activated, and it will reach to their target sites like conjunctival epithelium and respiratory epithelium of trachea and nasal cavity in this case, and ultimately it will again replicate at the site and it will produce disease again. So this is known as latency. So generally this latency is a characteristic features of herpes virus infections, and they are also produce latency in this case also. So generally it is said that whenever there is a herpes virus infection, in majority of the cases that particular animals or bird will infected throughout their entire life. Host, domestic fowl and occasionally pheasants are primary hosts for this disease. Generally all ages of birds are susceptible for infection, but majority of the outbreaks which occurs in birds are between 3 to 9 months of age. Generally in this case males are more susceptible than female ones. Now transmission. So generally air route is the most important, aerosol infections are most important route of transmission of this disease. As we have already discussed, this particular virus are replicate only in respiratory tract, respiratory epithelium of the trachea and nasal cavity. So this virus will replicate only in these organs, and whenever this particular aerosol routes are most important route of transmission of this disease. Ingestion of this virus can also responsible for production of the disease, but for that this virus must go into the nasal epithelium or tracheal epithelium for production of this disease. The another important route of transmission of this disease in case of infectious laryngotracheitis ILT is carrier birds and vaccinated birds when vaccination is given with the live attenuated vaccines. As we have already discussed, once birds and animals are infected with herpes virus, generally they become lifelong carrier, and in this case also this particular birds are become lifelong carrier. So there will be intermittent shedding of this virus into the environment, and ultimately they become source of infection for other healthy animals. Once animals are getting infected, whenever the immunity is developed, if animals survive from the initial infection, there will be immunity will be developed and there will be development of latency. So whenever such infected birds when go into the stress, or whenever there is immunosuppression due to any reason, in that case again this virus will get activated, and there will be shedding into the environment, so it becomes source of infection. Another important route is vaccination. Generally when attenuated live vaccines are used as a vaccines, in that case also there will be development of latency, and whenever there is immunosuppressions or whenever there is a stress to the birds, in that case also this virus will come out from the trigeminal ganglia and it will again shed into the environment and they become source of infection. So in both the cases, carrier as well as vaccinated birds, they become source of infection for other healthy birds. Another important thing that I want to share in this case also, that probably this ILT is the first viral disease, particularly avian viral disease, for that this vaccine was developed. So this is probably first viral disease for that vaccine was developed in past. Now we will see regarding pathogenesis. So we have already discussed that virus are generally replicate only in nasal epithelial cells, tracheal epithelial cells, which are both respiratory epithelia, as well as this particular virus is also replicating conjunctiva. So either through aerosol route or through ingestion route, first of all this virus will reach into the nasal epithelial cells or in tracheal epithelial cells. Initially there will be attachment and finally it will go inside the cells and ultimately it will start replication. The most important thing in this particular ILT is virus will generally infect one cells, it will again reach to another cells by cell to cell routes. Generally there will be no viremia occurs in this particular disease like other infection. So there is no exposures of virus to directly outside the cells. So generally there will be no exposure of these cells, particularly virus, to our host immunity directly. They will always remain inside the cells. So this particular virus will replicate inside the nasal epithelial cells, inside the tracheal epithelial cells, as well as they will replicate inside the conjunctival epithelial cells. And during this replication, it will induce degenerative changes as well as necrotic changes to the epithelial cells. So ultimately, due to this replication of this virus, there will be a degeneration, necrosis, and sloughing of a conjunctival epithelial cells, respiratory tracheal epithelial cells, and ultimately what you will find this particular entire slough materials are get accumulated into the tracheal lumen. So due to accumulation of this exudate, particularly fibrinous as well as necrotic materials as well as inflammatory cells which are get lysed, which are get degenerated, they all are get accumulated into the tracheal epithelial cells as well as nasal lumens. This all are get accumulated into the lumens of trachea. So ultimately it will occlude the nasal passage and there will be difficulty in breathing. So when if entire trachea get occluded, in that case there will be difficulty in breathing and finally there will be a severe dyspnea. At that time birds will try to open their mouth and you will find gasping, as well as sometimes this entire exudative materials, bloody exudative materials, sometimes they are come out from the mouth cavity. So you will find expectoration of mucoid, bloody mucoid discharge, and sometimes due to severe occlusions of the trachea you will find death. In some cases, if immunity is strong, in that case there will be, due to development of immunity, such type of birds will recover. But as we have already discussed, this particular virus will produce latency. So this particularly virus will reach into the trigeminal ganglia and this will produce latency. Why this particular herpes virus preferred nerves, particularly neurons for the latency? There are some factors which help for such type of behavior. First one, generally neuronal cells have no MHC class 2 molecules and very minimal numbers of MHC class 1 molecules are present over the surface. So generally there are very little chances that infected neurons will show their antigenic structure over the surface, and due to this reason this our this is particularly our immune cells will not able to identify such type of cells, such type of infected cells. So there are less chance of destructions of such type of infected cells. So generally it will produce latency, and after development of latency, whenever there is a immunosuppressions due to any reasons, whenever there is a stress to the birds, in that case there will be again reactivations of the infections occurs. This viral will get replicate inside the neurons. Ultimately it will come to again target sites like conjunctival epithelial cells, nasal epithelial cells, as well as respiratory tract epithelial cells of the trachea, and ultimately they will again replicate and it will produce disease again. Now clinical signs. Incubation period is range from 6 to 14 days. An infection may result as a poor acute, acute or mild form of disease or sometimes asymptomatic infection. So generally in per acute cases you will not find any type of clinical signs, suddenly you will find having mortality. In acute form generally you will find difficulty in breathing, gasping with head extended. You will find expectoration of bloody mucoid discharge. There will be cyanosis of face and vitals, and these all lesions are mainly due to, we have already discussed that this particular virus are replicate inside the tracheal epithelial cells. So ultimately due to the replication there will be necrosis and sloughing of the cells, and this all materials are get accumulated into the tracheal lumen, so it will occlude the tracheal lumens and there will be difficulty in breathing. So you will find gasping, open mouth breathing, difficulty in breathing, and this dead materials are which are get accumulated inside the tracheal lumen, they will come out from the mouth cavity and ultimately you will found bloody mucoid expectoration. In mild cases generally you will find conjunctivitis. As we have already discussed, this particular virus also replicate inside the conjunctival epithelium, so you will find conjunctivitis. You will find small swollen infraorbital and nasal sinuses. You will also find congestions of mucous membrane, and in case of layer birds you will find decreased egg production. So here you can see this majority of these gross pictures are taken from Cornell University website, as well as some of these pictures are taken from the NOHAS archives of C.L. Davis Foundation. So here you can see this is conjunctivitis. This entire periocular tissue are showing some sort of lesions. So they are showing swelling and excessive lacrimations from the eye. Here you can see these are the two birds are shown in these pictures. This one is comparatively healthy birds, while this is actually affected from the ILT. And here you can see this periocular tissue and conjunctiva are mainly affected. This eye tissue are showing severe conjunctivitis with redness, as well as this periocular tissue are showing some sort of exudate. They are attached with the skin of the periocular tissue. So here you can see this is another picture taken from the Cornell University website that are showing open mouth breathing, and this is known as gasping. We have all discussed why this gasping occurs, because this entire tracheal lumens are filled with the exudate. So generally you will find such type of clinical signs. In this case also you can see this is conjunctivitis. There is some sort of lacrimations, as well as some purulent discharge is coming from the conjunctiva. Now gross pathology. So we have all discussed that this particular virus is mainly replicate in conjunctival epithelial cells, nasal epithelial cells, and the respiratory epithelial cells of trachea. So lesions are also predominantly seen in this organ only. Generally other systemic organs are not much involved in case of infectious laryngotracheitis. So here what you will find, here this particular virus is replicate inside the cells and induce degeneration, necrotic changes. So generally what you will find necrosis, and this particular necrosis cells will get sloughed into the lumen of the trachea and nasal cavity, and this particular there will be severe infiltration of inflammatory cells at the sites. So you will find accumulations of infectious materials, fibrin exudations, as well as necrotic cells, and they all are get mixed with each other, and they will form some diphtheric fibrinonecrotic diphtheric membrane inside the tracheal lumen. So you will find necrosis, hemorrhage, and ulcerations. Generally you will find occlusive pseudomembranous or hemorrhagic cast or caseous plug in trachea. Many a time these materials which are present in the trachea, they will come out from the beak, and generally this soiled it will soil beak, face, or other part of the body. In milder cases you will find sinusitis, general infraorbital sinuses, they are many a time get enlarged. You will find conjunctivitis, you will find congestion of the mucous membrane, and many a time seromucoid discharge are come out from the eye. Generally, as we have already discussed, lungs and air sacs are rarely affected because this particular virus have very high affinity for upper respiratory tract epithelium only. So here you can see this is again, these pictures are taken from Cornell University website. Here you can see this is entire trachea are affected, and this entire trachea are slightly hyperemic, as well as they are also showing hemorrhages, multifocal to coalescing hemorrhages that involve entire trachea. Many times such type of lesions should be differentiated from other important disease like infectious bronchitis, Newcastle disease, and avian influenza. In this case also many times you will find such type of trachea. So, for in such particular like cases, you have to go for laboratory diagnosis for confirmation of the disease. But another important thing is, generally in case of ILT, generally you will find free blood tinged exudate in trachea, while such type of generally free materials are generally not present in case of avian influenza, Newcastle disease, and infectious bronchitis. Here you can see what we have already discussed. These are the pictures of the trachea, again taken from Cornell University website. So this is entire trachea are actually showing some pseudo-diphtheric membrane, and this pseudo-diphtheric membrane, they are nothing but they are sloughed necrotic epithelial cells, inflammatory cells, and mixed with the fibrin. And you can see this is entire tracheal lumens are covered with the such type of pseudo-diphtheric membrane. And most important thing, the differential diagnosis from other diseases, presence of such type of free exudate, such type of free blood clots or blood tinged exudate in tracheal lumens are generally indicative of ILT rather than infectious bronchitis, even in influenza and Newcastle disease. But still, they are suggestive of ILT. For confirmation of the disease, you have to go for laboratory diagnosis. Here you can see this is another picture. This entire trachea are filled with the caseous plug, fibrinonecrotic material. Here also they are filled with the fibrinonecrotic material. So, when you can see this entire trachea filled with the such type of material, so ultimately it will occlude the lumen, it will produce asphyxia, and ultimately birds will die. Here you can see this is again pseudo-diphtheric membranes are present over the trachea. All these materials which covered the internal layer of trachea are pseudo-diphtheric membrane. Now, microscopic pathology. So, generally in case of herpesvirus infection, generally you will find some typical lesions. First one is intranuclear inclusion body. Second one, syncytia formation, means formation of multinucleated cells from epithelial cells. And generally some lesions which are not seen in birds, and generally they are common in humans, particularly in viral herpes viral infections, are marginations of nuclear material, chromatin material, and molding of nucleus, right. So, generally in this case also you will find intranuclear inclusion body, but here you have to remember that these inclusion bodies are not always found. They are generally found in initial infection, when the cells are get sloughed into the into the lumen, and then after they will survive for a very few times, and only that particular time you will find typical intranuclear inclusion bodies, and generally this period is range from 1 to 5 days. So, generally you will find intranuclear inclusion body in early infection only, not in late infections or in chronic stage, because most of these sloughed cells will get die, they will get degenerated, and ultimately they will get lysed. So, you will not find intranuclear inclusion bodies. Generally these intranuclear inclusion bodies are found only in early infection between 1 to 5 days of infection. So, here what happen, this particular virus are replicated inside tracheal epithelial cells. So, ultimately there will be degeneration, there will be necrosis, and ultimately this tracheal epithelials will get sloughed into the lumen. So, you will find generally intranuclear inclusion body in sloughed epithelial cells. Then generally these lamina propria are generally severely infiltrated, or mucous are generally severely infiltrated with the inflammatory cells, as well as you will find some sort of congestion. You will find syncytia formation. Generally the epithelial cells, due to this viral cytopathic effect, these cells are get fused with each others, and ultimately you will find large multinucleated cells. But they are slightly different from macrophages, because they are macrophagic cells which are formed giant cells, multinucleated giant cells. But in this case epithelial cells are get fused with each other due to viral cytopathic effect, and they will produce to one large multinucleated cells. You will find lymphocytic, histiocytic inflammations, as well as many a time you will find also infiltration of heterophils in the mucosa and submucosa. This necrotic materials will get sloughed off, and due to this sloughing you will find ulcers, hemorrhages of the necrotic of the lining epithelium of the trachea. So here you can see this is beautiful picture, microscopic picture taken from C.L. Davis Foundation. So here you can see this is sloughed epithelial cells and into the lumen of the trachea, and these particular cells are showing typical intranuclear inclusion body. So this is nucleus. You can see this is nucleus. Some sort of margination of the chromatin material visible at the nuclear membrane, and you will find one large intranuclear inclusion body. This entire material is intranuclear inclusion body, right? So many of these necrotic cells are showing intranuclear inclusion body, as well as you can see this entire sloughed debris showing inflammatory cells, including lymphocytes, histiocytes, as well as heterophils. This all pink granules are actually heterophils. In case of birds, they contain such a bright pigments in the cytoplasm, and probably this is a syncytial formations. This is, you can say, this is multinucleated cells, which is again sloughed into the lumen. So diagnosis. So diagnosis of this infectious laryngotracheitis is based on clinical signs. Whenever you find bloody mucoid expectoration from mouth cavity, generally they are indicative of ILT. But again, this is just suggestive. For confirmation, you have to go for laboratory diagnosis. Generally, gross and macroscopic lesions, particularly gross lesions, whenever you find free blood tinged exudate in tracheal lumens and other, most of the other organs are normal, in that case you can suspect the ILT. But when you don't find such a free exudate in tracheal lumen, in that case such type of lesions should be differentiated from Newcastle disease, avian influenza, and infectious bronchitis. Microscopically, whenever you find intranuclear inclusion body, they are very characteristic features, and they are actually indicative of ILT disease. Intranuclear inclusion body are very characteristic feature of herpesvirus infection, but the main problem is they are seen between just 1 to 5 days of infections, and then after they are difficult to locate. So if you find, they are very characteristic and you can confirm the disease, but if they are not found, then you cannot rule out the disease, and for confirmation you have to go for some serological or laboratory investigations. So for that you have to go for virus neutralization test, agargel immunodiffusions, ELISA, PCR, and immunohistochemistry. On basis of that you can confirm the disease. One another test which is carried out in birds is generally for virus replication. Generally this particularly herpesvirus, Gallid herpesvirus type 1 are inoculated into the chicken embryo, particularly into the eggs and directly into embryonated eggs. So generally what happens, this particular virus will produce pock-like lesions into the chorioallantoic membrane. So here you can see this is chorioallantoic membrane, and you can see these white spots are actually plaques, and generally these plaques have a central depression, they are showing necrosis, while this periphery are generally showing hyperplasia of the epithelial cells. So generally this is the chorioallantoic membrane of the egg, and this all white spots are actually pock lesions, and these pock lesions are nothing but they have a central area of necrosis, and periphery you will find some sort of proliferative reaction. So such type of lesions are known as pock lesions, and these pock lesions are again generally indicative of virus replication, particularly Gallid herpesvirus replication. So this is all about infectious laryngotracheitis. Thank you.